We're going to start the parasitology unit with nematodes on chapter 45, page 284. Now, nematodes are another name for roundworms. So we're going to start our roundworm section with the superfamily Ascaroidea. And a more common name for Ascaroidea are Ascarids. So you can find these parasites and information on them on page 285 or 288 to 289 of your textbook. The Ascarids we're going to be covering in detail are Toxocara canis and cati, Toxascaris leonina, and Ascaris suum. Now in this lecture we're going to cover the morphology, hosts, life cycle, and pathogenesis of each of these parasites. In class, we're going to be discussing the treatments, prevention, and whether or not there is a public health significance for these particular parasites. Feel free at any time during this lecture to pause in order to write notes or write questions that you have about the material that's presented here and bring them to class with you so we can answer your questions in class. So we're going to start with Toxocara canis and Toxocara cati. Now I've coupled these two parasites together because they are actually the same parasite. They just infect a different species of animal. Now the adult worm of Toxocara is cream-colored, has cervical alae, and the cervical alae are these wing-like structures on the anterior end of the parasite, meaning the head end, and it also has 3 large lips, and these 3 large lips are located in about the same area where the mouth would be. The appearance of these adult roundworms are spaghetti-like, and you can find them in the small intestine. Now the larvae of Toxocara, you will find them in the liver, the lungs, and other tissues as they start migrating through the body in order to get to an adult. The egg of this parasite, also known as an ova, we will find these eggs or ova in the feces of the animals that have this parasite. The eggs will be spherical or circular. They'll be thick. They have a fine pitted outer edge. And what I mean by fine pitted, there are these little pits or little divots on the outside edge of the parasite egg. And they have a single embryo on the inside. Now the definitive host, or the main host for Toxocara canis is the dog, and Toxocara cati is the cat. Now there is a potential for an intermediate host. It is possibly a rodent, and we refer to the rodent as a paratenic host. A paratenic host is just a transport host. The parasite, if it is ingested by the rodent, the parasite does not die, nor does it develop to the next life stage. It's kind of like a waiting area for that parasite until the rodent is eaten by a definitive host, meaning the dog or the cat. Now the pre-patent period refers to the time when the infective stage of the parasite is ingested to the point where that egg or larva that had been ingested is starting to lay its own eggs. So the pre-patent time refers to the time when the infective stage is ingested by the definitive host to the point where it's actually starting to lay its own eggs or have its own babies, reproduce. That period is about 6 weeks. So you can see why this parasite is a major problem in puppies and kittens especially, because this parasite can reproduce rapidly. Now the life cycle of this parasite, it is a little intense to kind of interpret here. So I'm going to go through it with you just to give you an idea of how it all works. So the adults reside in the small intestine. They lay eggs, which are passed in the feces. Those eggs hatch and larvae start to develop in the soil. They go through a larva 1 stage or an L1 stage to larva 2 and to larva 3. Now once they get to the L3 or larva 3, that means they can be ingested by the definitive host and potentially mature to an adult. So the L3 is ingested by the definitive host, or the definitive host ingests the L3 through that paratenic host, like the rodent, or the L3 can also be passed transmammary in puppies to puppies or kittens through the mother. Now, Toxocara canis, they have found, can also be passed transplacentally, meaning that larva 3 stage can pass across the placenta into a puppy fetus still inside its mother. Now, once the larva has been ingested or is inside the body of the host, it undergoes extensive somatic migration, meaning it's migrating through the body, or it starts to migrate through the trachea. So it migrates through the body up to the trachea, where it can develop to sexual maturity. At that point, it's coughed up out of the trachea, it is swallowed down into the stomach, and heads to the small intestine, where it can develop into an adult. So, a couple of points to make here. Toxocara canis in the dog, tracheal migration is high in newborn puppies, but kind of fades out after 2 months of age. Toxocara cati, on the other hand, tracheal migration of this parasite can likely last the whole life of the animal if that animal is not treated. So, what kind of damage does this parasite cause? It can cause puppies and kittens to vomit or defecate worms, as well as adult animals as well. So vomiting, defecating the worms, you may see a distended abdomen or a pot belly appearance, as you can see in this bottom picture of the puppy. When puppies or kittens have a pot belly appearance, it's a pretty good indicator that they may have parasites, most likely this parasite, Toxocara, being very common. It may also cause colic in the animal, as well as intestinal obstruction or rupture. If worms are in the intestine in large amounts, like this picture in the right-hand corner, this is a serious issue. It can actually stretch the intestinal wall to the max and cause it to rupture if they get in that large of an amount in the intestine. It can be life-threatening, especially in puppies and kittens. Now let's talk about Toxascaris leonina, also known as the arrow-headed worm. The adults of this parasite are very similar to Toxocara. You find them in the small intestine. The larvae are also found in the small intestine as well as other tissues in the body as they migrate. Now, the ova, or the egg, for this parasite, which again you'll find in the feces of the animal, is oval in shape, oval. It is light-colored. It has a smooth outer shell, and just below that smooth outer shell is kind of a ropey appearance. So it's a little bit different from our Toxocara ova. Again, Toxocara has that fine pitted outer shell, and Toxascaris has that smooth outer shell with a thin ropey layer just under that smooth outer shell. Toxascaris leonina, again, very similar to Toxocara. It is in dogs and cats, possibly mice are the parotenic host or intermediate host in this case, and again the prepatent period is 6 weeks, just like Toxocara. Now, the pathogenesis of this particular parasite, you may have an animal experience unthriftiness and also that pot-bellied appearance again. Now, if you're not sure what unthriftiness means, it's basically a failure to grow or put on weight, even when that animal is fed quality feed in adequate amounts. So even though they're being fed enough and they're getting really good food with lots of nutrition in it to help them grow, they're just not growing or developing at the rate a normal puppy, kitten, or animal would in general. That's what unthriftiness is. Now, the last parasite I want to discuss with you is Ascaris suum. This is commonly known as the large roundworm of pigs and is probably the most common parasite in pigs. Now, the adults of Ascaris suum, again, they are white cream colored. They have 3 large lips on the scolex. And again, the scolex is like the head of the parasite. They're quite long, about 30 centimeters, and they're found in the small intestine of the pig. The larvae, again, they migrate through the body, through the liver, through the lungs in this case. And the ova that you'll see in a pig's feces is going to be oval in shape. It's got this yellowish brown, a little bit darker appearance to it. It's thick, and it also has a very bumpy outer shell, almost like a turtle shell, or I used to refer to this as piggy bumps. on the outside of this egg, and that helped me refer it back to its host, which is the pig. So again, definitive host, usually the pig. Occasionally, if a cow, sheep, or human ingests the egg of this particular parasite, they can become infected. There is no intermediate host, and the prepatent period is about 8 weeks. So in this life cycle, the eggs are passed in the feces, but the larva actually develops completely inside the egg. And the infective egg is then ingested by the pig as it's rooting around. The eggs hatch in the small intestine. Once they hatch, it proceeds to the liver through the portal vein. The larvae start to tunnel through the liver as they're molting and developing for a few days, and then travel out of the liver through the caudal vena cava to the heart, to the pulmonary artery, and then up to the lungs. All the while, they've caused this tissue damage in the liver. Then they start to migrate through the bronchial tree of the lungs, and they're still causing tissue damage. And then they head up to the trachea, where they're coughed up. They head to the pharynx, where they're coughed out, swallowed again, to the small intestine, and then they start maturing to an adult. So what does this parasite cause in pigs? Major tissue destruction, particularly in the liver and in the lungs. So we have milk spots on the liver, and again, this is from the tunneling larva. We can have hemorrhaging in the lungs, edema, which is fluid buildup in the lungs, and we can also see a lot of eosinophils, not just in the lungs, but also in the blood in general. Here's a picture of blood, and these little red round circles, these are red blood cells. This big circle here, this is actually a white blood cell, and a type of white blood cell is an eosinophil. Eosinophils are usually found in animals that, in high numbers, eosinophils are found in animals that have parasites or some type of allergy or allergic reaction going on. The animal that you are tending to may experience thumps, which is coughing, and heavy infections can lead to obstruction or perforation of the intestine. This bottom right-hand picture is the perfect example of an obstruction in the intestine and a potential perforation about to happen. So what I would like you to do is compare the ova of the ascarids that we just talked about here. You can see the fine pitted outer shell of Toxocara, the smooth outer membrane, and just slightly ropey inner membrane of Toxascaris, and that thick, bumpy outer shell, the piggy bump shell of Ascaris suum.